Microorganisms are present in the air and on surfaces within the lab environment. Because bacteria multiply rapidly, even a single unwanted cell can quickly overtake a culture. So transferring bacteria without contaminating your medium is a challenge. The set of procedures involved in manipulating bacteria without introducing contaminants is known as aseptic transfer. The transfer process involves moving a small amount of bacteria, called an inoculum, to a new sterile medium. Introducing an inoculum to a sterile medium is known as inoculation. Good aseptic technique begins with decontaminating your lab bench. This minimizes the amount of environmental microbes in your work area. Bacteria are transferred between culture media using an inoculating instrument. An inoculating loop, or simply a loop, is a metal wire that ends in a closed loop at the tip. Loops are used for a variety of culture transfers. An inoculating needle is also a metal wire, but it doesn't end in a loop. Needles are used in solid media inoculations that require penetration, or stabbing, of the medium. Before an inoculating instrument can be used, it must be sterilized to remove contaminating microbes. We'll do that with a Bunsen burner. First, attach the Bunsen burner to the gas supply, making sure the tubing fits snugly around the gas nozzle. The Bunsen burner has two important controls. The wheel at the base of the burner controls the amount of gas entering the burner. For maximum safety, begin with this valve closed. The amount of air entering the burner can also be controlled. On this burner, that is accomplished by rotating the barrel. To light the burner, first turn on the gas supply to the Bunsen burner. The handle should be parallel to the gas nozzle for maximum flow. Next, rotate the burner's wheel a half turn to allow gas into the burner. Hold the striker above and slightly to one side of the opening of the burner. Squeeze the striker to produce a spark that lights the burner. As you squeeze, make sure the flint of the striker scrapes hard against the abrasive inside. Rotate the barrel until the flame turns blue and a bright blue inner cone appears. The flame is hottest at the tip of this inner cone. If you're unable to light the Bunsen burner, turn off the gas supply and ask for assistance. Do not leave the gas on to an unlit burner. Sterilize the entire wire portion of your inoculating instrument by holding the loop in the hottest part of the flame. Make sure the entire wire gets red hot, not just the tip. Allow the instrument to cool for 15 to 30 seconds. Never touch a hot instrument to any culture. Not only will this kill bacteria in your inoculum, but it can create an aerosol that spreads bacteria into the air. Once properly flamed, the inoculating instrument is sterile. While it's cooling, don't recontaminate it by touching the metal with your hand or setting it on your bench. If you need to set the instrument down, use a rack or a stand. First, we'll obtain bacteria from a broth culture and use it to inoculate sterile broth in another tube. Begin by labeling the sterile tube with the identity of the inoculum, your name, and the date. Flick the culture tube to suspend the bacteria. Be careful not to spill the contents. Notice the sediment in the tube moving into suspension. This will ensure that your inoculum contains a sufficient and representative sample of the bacteria. Hold the flame-sterilized loop like a pencil. Hold the test tube at a slight angle with your other hand. Do not tilt the tube too much, or broth and bacteria will spill out. Remove and hold the cap with your ring and pinky finger of the hand holding the loop. Never set caps on the bench top. Pass the mouth of the tube through the flame, quickly. The hot air will keep airborne contaminants out of the tube and kill any bacteria at the mouth of the tube. Use your loop to obtain a loopful of broth culture. As you remove the loop, be careful it doesn't touch the walls of the tube. While holding the loop steady, pass the mouth of the culture tube through the flame again and recap. This second flaming kills any bacteria that may have settled on the mouth of the tube while it was open. Return the culture tube to the test tube rack. Now pick up the sterile broth tube that will receive the inoculum. Remove the cap. Flame the mouth of the tube. Insert the loop containing the inoculum into the sterile broth and gently swirl the loop. Then, remove the loop from the tube, taking care not to touch the loop to the side of the tube. Flame the mouth of the tube and replace the cap. After returning the tube to your test tube rack, flame the loop to destroy any remaining bacteria. Make sure the entire wire gets red hot, not just the tip. Now let's see how this procedure differs when transferring broth culture to an agar slant. We've already seen the steps for removing a loop full of culture from a broth. Flame the mouth of the broth tube and recap. Return it to the test tube rack. Now pick up the agar slant tube that will receive the inoculum. Remove the cap. Flame the mouth of the tube. To transfer the loop full of culture to the agar, hold the loop parallel to the agar surface. Touch the agar and then gently zigzag the loop over the surface while withdrawing the loop from the tube. Do not dig into the agar, or you will deposit most of the bacteria inside the agar instead of on the surface. Flame the tube and replace the cap. After returning the tube to your test tube rack, flame the loop to destroy any remaining bacteria. Slant cultures form a solid growth streak on the surface of the agar, so the density of bacteria is much higher compared to a broth culture. To obtain an appropriate amount of inoculum from the slant, simply touch the edge of the loop to the growth streak. Then lift the loop from the growth and remove it from the tube. Do not drag the loop over the culture surface. When transferring the inoculum from the slant to a sterile broth, the solid culture tends to stick to the loop. So swirl the loop gently within the broth to dislodge the inoculum. Be careful not to touch the loop to the side of the tube. Certain biochemical tests require inoculation of bacteria by stabbing into an agar deep. This procedure requires an inoculating needle. Sterilize and cool the needle as you would a loop. A solid culture, such as that on a slant, is usually used to inoculate deeps. Remove the cap of the culture tube, then flame the mouth of the tube. Insert the needle carefully and touch the side of the wire to the growth. Be careful not to stab into the agar. Gently roll the needle from side to side on the growth surface to pick up some of the growth. Then remove the needle from the tube. Flame the mouth of the culture tube, then recap and return it to the test tube rack. Now pick up the agar deep, remove the cap, and flame the mouth of the tube. Stab the agar in the center of the tube, penetrating the needle about 2/3 of the agar depth. Withdraw the needle along the same path as you stabbed. Flame the mouth of the tube, recap, and return the tube to the test tube rack. Flame the needle to destroy any remaining bacteria. After isolating colonies on a Petri plate, you may wish to transfer a portion of that colony to a slant for long-term storage. This aseptic transfer is similar to the transfer from an agar slant, with a few minor changes. On the bottom of the plate, circle and label the colony that will be the source of your inoculum. Also label the agar slant with the date, your initials, and the identity of the colony. Sterilize and cool the loop. Lift the lid of the Petri plate just enough to allow you to insert the loop and pick up a small portion of the colony. This technique prevents airborne bacteria from falling onto the agar. Withdraw the loop, taking care not to touch the Petri plate, and close the lid. While holding the loop steady with one hand, use your other hand to pick up the agar slant. Remove the cap, flame the mouth of the tube, and carefully insert the loop into the tube. Holding the loop parallel to the agar, gently touch the agar surface and zigzag the loop along the surface toward the opening of the tube. Flame the opening of the tube, recap, and return the tube to the test tube rack. Flame the loop to destroy any remaining bacteria. After you've finished your inoculations, remove all materials from the benchtop, making sure to discard any contaminated materials in the appropriate waste container. Then decontaminate the benchtop once again